Thank you everyone from around the world for joining us today. My name is Lorraine Rush, Vice President of Scientific Strategy and Commercial Development at Celerion. Today we'll begin with some quick housekeeping items. The estimated time of our presentation today is 30 minutes plus time for Q&A. Remember to submit questions via the Q&A button, and we'll address as many of those as time permits. Any questions we don't get to, we will actually reach out to you from Solarium directly within 24 to 48 hours. I'm very excited to share the new developments around our future of pharmacy expansion at Celerion. As many of you may have read in our press release, this capability allows our clients to work with us under the new guidelines for handling investigational product in accordance with the appropriate USP designations. Today, we're fortunate to have Kate Reese sharing her pharmacy and regulatory expertise to help position you, our clients, towards successful early clinical trial dose preparation. Kate received her Doctor of Pharmacy from Skaggs School of Pharmacy, University of Montana. Managing the clinical pharmacy responsibilities for over a decade at one of the largest clinical research units in the country, she is an expert in all aspects of IP compounding. Kate has played a pivotal role in the design and execution of the multi-million-dollar pharmacy upgrades for Celerion, ensuring that this industry-leading pharmacy meets all of the evolving regulations under the current guidelines. And with this, I turn it over to Kate.
Thank you, Lorraine, and thank you everyone for joining. My goals for the presentation today is to provide an overview of the challenges and trends that we are seeing in early clinical research, to highlight key elements of a basic phase 1 pharmacy checklist, to discuss the differences between good manufacturing practices and extemporaneous compounding and what is actually needed for an early phase clinical study, to discuss emerging USP and FDA requirements with regard to pharmacy compounding, and to give an overview of pharmacy services offered at Celerion's early phase clinical units. Phase 1 studies aim to gather information about a drug's pharmacology and its short-term safety and tolerability. There can certainly be challenges with regard to drug product when first starting clinical studies. Some challenges that may be encountered are, first, in some instances, there is a limited amount of drug substance available, which can present issues when trying to plan clinical development strategies. Next, prior to first-in-human studies, typically very little formulation development has taken place and minimal manufacturing processes are established at this stage. But there's also a real desire to obtain data in humans as soon as possible to guide clinical development decisions. As far as trends go, we are increasingly seeing more complex and adaptive study designs. There is also a trend towards highly potent compounds. These compounds require special handling processes and environmental controls, which I'll go into a bit more later in the presentation. We are also seeing an increase in the number of studies in which the active pharmaceutical ingredient, or API, is compounded at the clinical site for administration to participants. In moving from preclinical studies to phase 1, there are some requirements that need to be taken into consideration for the drug product. First, you need a dosage form that can be easily administered to humans. You also need a dosage form that allows administration of small doses, which are required for first-in-human studies. The doses for these studies are typically much lower than the anticipated therapeutic dose. You need stability data on the drug, which covers the duration of the investigation. And lastly, all components of the dosage form need to be GMP grade, including the drug substance and all excipients. Now, just because the API and the excipients must be GMP quality, that does not necessarily mean that the final dosage form itself must be produced under GMP conditions. To look at this further, let's discuss the differences between good manufacturing practices and extemporaneous compounding. Good manufacturing practices, as it relates to drug products, are defined differently in different parts of the world. In the United States, for drugs, GMP means the production or processing of a drug product following set quality standards for the purpose of distribution or resale. Extemporaneous compounding, on the other hand, is defined as the preparation, mixing, assembling, and altering of a drug in accordance with a licensed practitioner's medication order for a specific individual. The key difference between the two, in the context of clinical research in the U.S., is that compounding is done under the delegation and direction of the principal investigator and the protocol. It must be overseen by a licensed pharmacist, and the dose is prepared for a specific subject. Manufacturing is the bulk preparation of the drug product for distribution or use by a third party, such as a CRO or a doctor's office, and not for immediate patient or participant administration. And that's really an important key difference. Is the drug product being prepared for a specific participant, or is the product being produced in bulk to be distributed for later administration to an unknown study participant? Given these two different processes, let's look at what is required by the FDA for compounded drug products. As part of an IND filing with the FDA, the sponsor must include a section which describes the composition, manufacture, and control of the drug substance and drug product. The information included in these sections needs to be appropriate for the investigations covered by the IND, but the agency recognizes the amount of information needed will vary based on the phase and duration of the investigation and the amount of information available. The FDA also recognizes that the information included is likely to change as clinical development progresses. For example, changes in the method of preparation or the dosage form, or even the dose itself, are very likely to change throughout development as more information is learned. Final specifications for the drug substance and drug product are not expected until the end of the investigational process. The required sections of the IND remain the same regardless of the phase of the investigation or whether the final dosage form is produced under GMP conditions or is extemporaneously compounded. The sponsor needs to include a description of exactly what and how much is being administered, how and why the drug product is being prepared in the manner described. The FDA is not only interested in what you are administering, it is also important to describe why you are administering what you plan to. The sponsor needs to include specific information that provides evidence that ensures you are administering what you say you are. So your acceptance criteria and analytical procedures for the drug should be detailed. And lastly, stability information. Stability data is required for all phases of drug development. Information included needs to provide evidence that ensures the quality of the product for the duration of the investigation. However, for short-term investigations and compounded products, the stability data may be correspondingly limited. So to reiterate, the information included serves the same fundamental purpose regardless of whether one is compounding a new drug substance for a first-in-human study under an IND or whether one is selling a drug under an NDA, and that purpose is to provide scientific evidence that the drug product is produced to the highest quality standard possible for the safe use in humans. This is an example from the drug product section of an IND accepted by the FDA for one of our sponsors for an extemporaneously compounded oral solution with information updated or redacted for the purposes of this presentation. Simply stating where the product will be compounded and providing a description of the compounding procedure is generally sufficient in most cases. Again, using the same example, this is the specification information that was included. In this case, the release criteria for the solution was visual inspection, with the acceptance criteria clearly stated as a clear or slightly hazy homogeneous solution. As previously discussed, stability data needs to cover the duration of the investigation, but with compounded products, the stability data can be limited to the in-use period for the drug product. In our example, it is stated that the in-use stability data covers 6-hour use period, which means the solution must be prepared and subsequently administered within 6 hours. This is something that is easily doable with an on-site compounding pharmacy. Going back to the Phase 1 pharmacy checklist, again, we need a dosage form that can be easily administered to humans and provides the ability to administer small doses that are required for first-in-human studies. On-site pharmacy compounding allows for preparation of small formulation batches and for dose customization. Examples of some formulations that can achieve these requirements are neat API in a capsule or an oral solution or suspension. We've already discussed that GMP quality ingredients are needed, but that the final dosage form does not necessarily need to be produced under GMP conditions for early Phase 1 studies. You do need stability data for the selected formulation. However, for compounded products, which will be prepared and administered in a relatively short period of time, the stability data can be correspondingly limited. Typical in-use stability information for compounded products is generally limited to hours or days, rather than months for a standard GMP manufactured batch. I think there are some clear advantages in utilizing extemporaneous compounding for early clinical studies rather than completing a full manufacturing run. Starting with timing. Traditional manufacturing can take several months for batch production and release, whereas compounding requires minimal lab and stability information to get up and running and can typically be completed within a few weeks. Cost is significantly different between the two options. Manufacturing can cost upwards of tens of thousands of dollars. On-site compounding is typically a fraction of that price. Last, and in my opinion, probably the biggest advantage, is the flexibility allowed by compounding. For your first-in-human studies, where you really don't know where the data is going to take you, compounding allows you to make adjustments to the dose level, or even the formulation for that matter, which is guided by real-time study results. You are not limited based on the formulation and dose strength available of the manufactured product. I don't know how many times we have seen data come back from the first cohort or two of a single ascending dose study that didn't follow the predicted models, and we've needed to modify the planned dose level in order to proceed. On-site compounding really does allow you the flexibility to make adjustments quickly and efficiently while preserving your study timelines. In the United States, pharmaceutical compounding guidelines are issued by the United States Pharmacopoeia, or USP. It's important to note that while the FDA does not specifically issue these standards themselves, the USP guidelines for compounding are enforceable by the FDA, meaning they must be followed. There are several major chapters in the USP compendium that impact pharmaceutical compounding, and I'd like to discuss a few. First is the USP Chapter 795, which provides standards for compounding non-sterile preparations. The second is Chapter 797, which outlines the minimum standards to be followed when preparing compounded sterile products. Listed here are examples covered by each chapter. In June of last year, USP published revisions to Chapters 795 and 797. These revised chapters are not yet in effect, but they are forthcoming, and there are some notable changes that I would like to highlight. Starting with the compounding facility. Areas related to compounding must now be separate from areas not directly related to compounding. With this revision, gone are the days where compounding can take place in the middle of the pharmacy with other activities occurring in the same vicinity. The facility needs to have dedicated compounding spaces, and these spaces must be designed, arranged, and used in a way that prevents cross-contamination of the workspace and drug products, and also promotes conditions for subject safety, worker safety, and environmental protection. Depending on the nature of the product and the dosage form, additional primary and secondary engineering controls and equipment may be necessary. For example, all powder manipulation, such as weighing and measuring, must occur inside a containment device, such as a biological safety cabinet. A safety data sheet and a certificate of analysis are now required to be on file for every API received by the compounding facility. Another important update to Chapter 797 is the change from low, medium, and high-risk compounded sterile products, or CSPs, to Category 1 and Category 2 CSPs. The main difference between Category 1 and Category 2 products is the environment under which they are prepared and the beyond use date or product expiration assigned to the compounded preparation. Both Category 1 and Category 2 must be prepared within a HEPA-filtered laminar flow hood or a biological safety cabinet. For Category 2, which is by far the vast majority of sterile preparations, the hood must also be placed within a HEPA-filtered clean room. There should be routine environmental monitoring of the compounding spaces to ensure that the primary and secondary engineering controls are functioning properly in order to prevent biological and cross-contamination of the drug products. Revised Chapter 797 requires an increase in the frequency of biological air and surface sampling to ensure that the compounding environment is maintained and suitable for preparation of sterile products. Another major revision to the chapters is the updated requirements for personnel training and competency assessments. Compounding personnel need to be qualified to perform their duties and need to be reassessed in those duties semi-annually. These assessments need to be completed not only in written format, but also didactic testing where the personnel actually perform and are evaluated on the respective tasks that they are authorized to complete. USP revisions will require that each facility name a designated person or persons who is responsible for ensuring compliance with SOPs and USP guidelines. The updated chapters also place a major emphasis on the need for the compounding facility to have an implemented quality management system. All of the processes described are vital to ensure that the compounded products are produced to the highest quality standard possible. However, when something does not meet specification, the compounding facility needs to have an established process to investigate, track, and trend quality issues and implement corrective and preventative actions when appropriate. Another important update from USP is the issuance of Chapter 800. This new chapter went into effect December 1st of last year and provides standards for safe handling of hazardous drugs to minimize the risk of exposure to healthcare personnel, patients, and the environment. Pharmacies should be completing a documented risk assessment for each compound that enters the facility to determine appropriate containment strategies and work practices. The NIOSH criteria is used to determine whether or not a drug poses a hazard risk. If insufficient information is available for the compound to make a determination, the drug is to be treated as a hazardous product. The vast majority of drugs in phase 1 studies fall into the category of insufficient information available and therefore need to be treated as a hazardous compound with appropriate containment strategies determined by the pharmacist or as outlined by USP. USP 800 standards apply to all healthcare personnel who receive, prepare, administer, transport, or otherwise come in contact with hazardous drugs and all environments in which they are handled. 800 requires that facilities handling hazardous drugs have designated areas for receiving, storage, preparation, and compounding. The designated areas need to be negative pressure to the adjacent or surrounding areas and need to be externally vented. Compounding or manipulation of the hazardous drugs must also be completed within a containment primary engineering control, or CPEC, such as a biological safety cabinet, which again must be located in the dedicated negative pressure compounding area. These new environmental control requirements affect nearly all pharmacy practices across the United States and have precipitated many pharmacy remodels, including at each Celerion U.S. clinical site. Chapter 800 also outlines the need for each facility to have a dedicated hazardous communication plan with specific training requirements for all staff who may come in contact with hazardous drugs prior to staff performing those job functions. Lastly, Chapter 800 provides standards and minimum frequencies for the cleaning of the environment, equipment, and devices used for hazardous drug handling and compounding. These items must undergo chemical deactivation when possible, decontamination, and then be cleaned for sterile compounding areas. Equipment and devices must then subsequently be disinfected as well. There's a lot to consider when evaluating compounding facilities for early phase clinical studies. First, you want to ensure that the facility has the dedicated compounding areas with appropriate engineering controls, whether that be the negative pressure externally vented rooms and hoods for preparation of API and hazardous or potentially hazardous drugs, or appropriate clean room environments and laminar flow workbenches for the preparation of sterile products. Secondly, the institution needs to have robust standard operating procedures detailing not only drug handling and compounding processes, but also environmental maintenance and cleaning requirements. The facility should have established quality management system that details quality investigation and corrective and preventative actions. And lastly, the facility is only as good as the people working there. The site needs to employ trained and knowledgeable staff. Extemporaneous compounding in the U.S. is required to be overseen by a licensed pharmacist. You also want to make sure that the staff is up to date on their training and continuing education requirements, and that ongoing study and compound-specific training is being completed. That leads me to Solarion Pharmacy Services. Each Solarion clinical site has a secured, limited access on-site pharmacy. The pharmacies are equipped with continuous environmental monitoring for all drug storage and preparation areas. Drug storage areas include controlled room temperature, refrigerated storage, and both -20 and -80 degree freezers. U.S. pharmacies are DEA licensed and can handle all controlled schedules, including Schedule 1. Solarion pharmacies follow global SOPs, which detail processes for all IP handling, preparation, compounding, drug retention, and drug disposition, as well as equipment maintenance, cleaning, and training. So if you are running one study in our Phoenix facility and one study in our Lincoln, you can rest assured that our processes are global and there will be consistency between the sites and the studies. All drug is prepared and dispensed on a unit dose basis and verified by 2 pharmacy associates. Each dose dispensed is labeled with a unique Solarion ClinQuick barcode label. This drug package label and barcode correspond to the specific subject for which the dose was prepared. At the time of dose administration, the clinical doser will scan the drug package barcode and scan the subject's wristband barcode, ensuring that the correct dose is administered to the correct subject. Additionally, Celerion has a global quality management system that is overseen by the Quality Assurance Department for the investigation, tracking, and trending of quality issues and the implementation of corrective and preventative actions as needed. While this presentation has heavily focused on extemporaneous compounding thus far, Solarion does have the capability to receive, prepare, and dispense investigational products in a variety of forms, from traditionally manufactured tablets and capsules, solution suspensions, topical formulations such as patches or creams and gels. It's also standard for us to conduct studies with ophthalmic products and inhaled or nebulized products as well. All Solarion pharmacies do have the capability to perform both non-sterile and sterile compounding, including compounded oral solutions and suspensions, API and capsule, as well as injectable, inhaled, and infused sterile products. Solarion has the ability to source various excipients and concomitant medications on behalf of the sponsor for on-study use as well. The Solarion Clinical Unit in Lincoln, Nebraska also offers ADME and microtracer services, which typically involves the pharmacy compounding of the radio-labeled test material. We have an authorized nuclear pharmacist on staff who is heavily involved in the setup, oversight, and execution of these studies. In light of the newly issued and forthcoming USP revisions, Solarion has invested in facility upgrades at both U.S. clinical sites. Each site will have a custom-designed USP-compliant compounding suite. The suites are comprised of dedicated labs for the preparation of sterile and non-sterile products for both hazardous and non-hazardous drugs. Additionally, the Lincoln facility offers a dedicated ADME lab for the preparation of radio-labeled products. These dedicated suites are certified to meet or exceed USP environmental standards by an outside vendor every 6 months. Each suite is equipped with a variety of dedicated, specialized equipment for preparation of compounded products. Each lab has a biological safety cabinet, with the cabinets in the hazardous drug compounding suites being externally vented. We have a variety of balances available, including both analytical and microbalances, in addition to other standard and specialty compounding equipment. Solarion is also undergoing extensive SOP and training program revisions to align with the updated guidelines. While USP has not announced a date when these revised chapters will become official as of yet, Celerion has moved forward with the pharmacy renovations ahead of the implementation of the guidelines. Construction at the site has been staggered. The Lincoln compounding suites were commissioned and have been in operation since February of this year. Phoenix renovations are currently paused as part of our COVID-19 risk mitigation plans, but are anticipated to be completed by early next year. This is the floor plan of the Lincoln compounding suite. As you can see, there are 4 dedicated labs for the preparation of compounded products, as well as dedicated hazardous drug storage areas as required by USP. The Phoenix compounding suite will have a similar layout with the exception of the ADME lab. The compounding process begins with the sponsor providing an outline of the preparation procedure or a pharmacy manual. From there, the Solarian pharmacist will draft a master formulation record and provide to the sponsor for review. Once the master formulation record has been approved, a mock preparation may be completed at the discretion of the sponsor or pharmacist. We find that mock preparations are particularly useful for complex procedures and allow for adjustments to be made to the preparation process and master formulation record prior to study execution, which ensures that on-study compounding proceeds seamlessly. Mock preparations are typically completed within 2 to 3 weeks prior to the first dose of the study. After the mock preparation is complete, any adjustments to the compounding procedures and master formulation record are made. Then on-study compounding and study execution takes place. Compounding and drug administration are completed within the in-use stability timeframe for the product, which again typically occurs within hours or days of product administration. All compounding is overseen by one of our staff pharmacists, and each compounded dose prepared is labeled with a Solarium subject-specific ClinQuick barcoded label to ensure the accuracy of the dose administration. Examples of preparation completed by Celerion include API in capsules, which we have filled with weights as low as 0.2 milligrams, non-sterile to sterile IV solutions, oral solutions and suspensions or powder in a bottle, as well as a variety of different taste masking and formulation blinding. Our pharmacies are staffed with both dedicated pharmacists and pharmacy technicians, with our team of pharmacists having over 25 years of combined phase 1 clinical research experience. A lead pharmacist is assigned to every study and will be your main point of contact for all pharmacy and compounding needs. The pharmacy staff complete a comprehensive training program inclusive of both internal and external pharmaceutical compounding education, hazardous drug training, as well as ongoing study-specific training and professional continuing education. We at Celerion pride ourselves on being your pharmaceutical compounding experts and are continually making investments in our service offerings to further support the needs of our clients, as demonstrated by the investments made into the USP-compliant compounding suites at our early phase clinical units. At this time, I would like to thank you again for joining today and invite you to reach out to myself or one of our dedicated pharmacists for more information on Celerion pharmacy services. And if there are any questions, I'd be happy to answer them at this time. Thank you.
Thank you, Kate. That was really, really interesting. We have a couple of questions that we have time for. We'll review the first 4. And the first one is, what are some of the sterilization procedures that Celerion has on site?
So sterilization procedures, as far as compounded sterile products and sterilization for participant administration, Celerion's standard process is to do sterilization by filtration, utilizing a sterilization grade filter that is suitable for the actual drug product. We do have a variety of sterilization and cleaning procedures, as well as on-site autoclave units for the sterilization of equipment and tools prior to preparation. But for the ultimate compounded preparation sterilization, we do utilize sterilization by filtration.
All right, thank you. Number 2, could you remind us how many compounding spaces are available at the Celerion pharmacies?
Absolutely. So the Lincoln, Nebraska facility has 4 dedicated compounding suites. The Phoenix facility will have 3. The difference between the 2 sites is that Lincoln has our on-site authorized nuclear pharmacist and the capability to perform ADME studies and microtracer studies. Otherwise, both sites have the same capabilities, including sterile preparation for both hazardous and non-hazardous products, as well as non-sterile compounding, of both hazardous and non-hazardous products.
All right. Another question is, how are these evolving regulations similar or different to the QP process in the United Kingdom?
That's a really good question. So what Solarion does have a facility in the Belfast, Northern Ireland, which does follow the European GMP and QP process guidelines. Manufacturing the definition of manufacturing differs between the 2 areas. In the United States, we consider the on-site preparation for our in-house study participants falls under the compounding guidelines. That same exact procedure would follow under manufacturing guidelines in Europe. The differences, honestly, there's less kind of paperwork to jump through. In the U.S., we rely on the certificate of analysis of the excipients used in the compounded preparation as evidence of release for the drug product, where in the EU, they need the formal review and release of that product by a specific qualified person and QP. In the US, that responsibility falls on the pharmacist, and those procedures are documented in our compounding record, which become part of your study file for the compounding of the drug product for your study.
All right. And one final question, what is the minimum time needed to adjust doses between cohorts in an adaptive study?
That is another really good question. So when utilizing compounding, really we have a lot of flexibility. I would say our standard turnaround from having the safety review team meeting and dose determination to being able to adjust and update, from a planned dose level, it typically is about within 24 hours. A meeting on day -2, compounding for the study on day -1 with dosing on day 1 would be a standard timeline for us. We can certainly condense those timelines if needed, and depending on the drug preparation process. Although sometimes, we have run into situations where, they want to switch to a micronized formulation of their API. And those are the sorts of things that, take a little bit more time. However, having the on-site pharmacy being in the same clinical unit where your study is being conducted really can streamline, in the example of the micronized situation, they were able to send out drug, pharmacy was able to receive it and prepare it for the next day. It really took less than 3 or 4 days to make that adjustment and move forward with the study.
Well, it's really very informative. Thank you, Kate. This concludes the webinar. We look forward to continued conversations with you. Our clients, a copy of this webinar will be available on our website. And as a reminder, any outstanding questions will be addressed by Celerion within the next 24 to 48 hours. And I'd like to thank you, our clients, and Kate for your time today.